So I'll do a brief introduction. Today we have a special session of our Design Learning Guild, and it's also brought to you by the CIRCL, the Center for Innovative Research on Cyber Learning Center. Dr. Ingmar Riedel Kruse is an assistant professor of bioengineering at Stanford, and his lab focuses on multi-cell biophysics and interactive biotechnology. He received his diploma in theoretical physics at the Technical University Dresden and did his PhD in experimental biophysics at the Max Planck Institute for Molecular Cell Biology and Genetics, followed by a postdoc at Caltech. And we're delighted you're here to tell us about interactive biology, cloud labs, biotic games, do-it-yourself kits, and more.
Good. So, welcome. Thanks for coming. So what I want to talk about today is one part of my lab. It's about a general idea of how can we as layperson or also as professionals interact with the small things that we usually don't see, like the cells and other biological material. And we do this for a variety of purposes. Education is one of them, and I think it's the more easy one to approach. But there's also some more serious things that we want to tackle, like research and others. So just for very general comparison, so if you look to the left, that's what computers were like 50, 60 years ago, right? Like big machines that filled rooms, were hard to operate, were not very powerful, and what we have today is like these interactive, intuitive devices that even small children can play with, right? And so these machines were for number crunching. This is really human experience, and if you now look at biotechnology, what's going on there today, we can actually draw many parallels to 50 years ago in computer science. So for example, here we see a microfluidic chip, which is used for diagnostic. It's lots of little, it's a device where you can basically do diagnostics, manipulate cells, biological materials, and so forth. And there are many analogies, like you have equivalency of a transistor and so forth. And so the questions that we have is like, could we do something similar like that in the future that you see in the background? Like, could we really interact with those things in a more playful way and not just having laboratory experts working with that? And so this brought me initially to this idea, let's play games, which is maybe one of the most intuitive thing from an educational, approachable point of view. And so what we set out to do is to build something that really relies on biological processes and also that is enabled by modern biotechnology. And to make this clear, it should not be just simulated like a regular video game, and it should not be horses or something which is more macroscopic, which we're used to, which we could call ancient biotechnology, right? And so one example that we built early on, which gives a good prototype and paradigm of what we're meaning here is the following. So you see these cells, these are paramecia, single-celled organisms, rather big, and if you have them in a microfluidic chip, with a few electrodes, we can actually use the galvanotactic properties. We apply mild electric fields in real time, and that makes those cells move around. And so you see this kind of gamepad here as an inlay. This is the computer screen online. And so when you push a button, the cells start moving, and you have virtual objects here where I try to make the cells swim through and thereby generate goals, and you can really play with that, right? And you can call this an experiment, but you can also call this a game. And so with the same kind of game engine, so to speak, you can make different types of games out of it by just programming different virtual objects on top of that, right? So for example, playing the soccer game here. So this was kind of our very early prototypes, as you see here, with a simple webcam, simple microfluidic chips and so forth. We also published this. We got good responses both from the scientific community as well as the more geeky game environment. And so this really made us think there is something here to bring these things more forward, make them more robust, and really use them for public engagement, for education, and maybe also for research. And so now in the talk, I want to walk you through a few projects where we made advances towards this. So in the second one, we actually use a different microorganism called Euglena. It's a single-celled organism. It's phototactic, and we built a device here, developed a device which is a microscope where you can play with, you can put your cell phone on it. And the idea behind is that a student can build the whole thing, learn about building and fabrication, but at the same time do experiments with it and play and kind of get all these things combined. And this is really motivated if you look at other areas, especially in mechatronics, right? There are all these robotics competitions. And in life science, we're really envious seeing what kind of neat toys and cool stuff they have there. And I think we're kind of really lacking on this in the life sciences. And similarly, more recent in the computer sciences, there's, for example, this National Video Game Challenge, which really is the idea of enticing children to program games and thereby to learn about computer science, to learn programming. And so we want to have the similar things in the life science, right? So here you see a video of these single-celled organisms, Euglena. So they're about 50 micrometer in size. So you can't really see them by naked eye. You almost could. They usually live in ponds, and they're very often used actually in regular school education. So they're also very easy to get from a school supply company. And another nice feature is that you can put them in a closed vial on your desk for a couple of weeks, and they're still fine. So this really makes it robust, anything you want to do with them. And just a little bit more about the phototaxis of these organisms, because that's what we are interested in using for all our interactive experiences here. So again, you have the cell, you have this red eye spot, which is actually sensing the light, and you have this long flagellum, which actually propels the cell through the fluid. And so the cell basically has this one eye spot, so this one-dimensional way of sensing its environment, and what's then doing is kind of rolling around its own axis, thereby sensing its 3D environment. And with a closed feedback loop between the sensory stimulus that falls onto the eye and this flagellum changing its beating behavior, it's then able to swim to the light or away from the light depending on what it needs to do. And what it usually does is, like, for very low light condition, it goes to the light. For very high light condition, it goes away, right? So we can basically use this in a very simple way to make an interactive experience out of this, where we have a little chamber here with 4 LEDs, and depending on which LED you shine on from one side, all the cells are swimming in a certain direction. So pretty similar like with these earlier paramecia that I showed you. And so here's a video of what this then looks like on the phone screen. So this is the cell phone screen. You see here the live view of these cells. And if you now turn on the light, then the cells start swarming into one direction, right? And there's a live tracking of one cell here, and so you can basically now try as a player to get this virtual ball somewhere to the other side. There's a number of other things here. For example, we bring this little cell close up here. If you look more closely, you can see subcellular details like the eye spot and other things. There's also the speed and the scale down here. And so there's a number of features that you see as a player that really are relevant for the learning context, so to speak, right? So ideas, while you engage and interact with these systems, you learn something, right, in a rather playful manner. So just what I said, right, like there's a number of things, of design features we can put on the screen, and there's presumably more we can do. There are really two major ways of how to interact here. One is you really tab on the screen and select a particular cell that you want to interact with, and the other is you use a joystick to make these cells move. And the whole thing can be built. So this is basically the whole building plan. So you 3D print your microscope. There's some simple lenses and eyepieces to put this on. There's some rather simple electronics, also with some double-sided tape. You can make these fluidic chambers. And so while building this whole setup, which, if you have all the material ready, not taking printing time as well, takes you 2 to 3 hours. And while doing so, you really engage with a number of STEM areas, right, like electronics, optics, and so forth, and really can engage in those and learn those. And in the end, have the gratification of having built something that you can use. You can also do experiments, right, like not just play games, but do formal experiments. For example, where you turn different lights in different directions, have a recording, and afterwards see what the traces look like, analyze that. You can also, so we programmed some applications where you can measure the speed for cells under different conditions a number of times. And so you can, for example, see as a student of does the cell swim faster when the light is on versus not, right? And then you can also think about programming, and so the Android language that all of this is living in is rather challenging to do. And so we made some simulation of that with the Scratch children learning language. And so you can basically recapitulate with very easy language what you kind of see there in the natural environment. And actually, the code here is all you need to kind of animate the light response of such a single organism. And so it's not only about learning programming, but this is actually already biophysical modeling, right? Like making a model of the cell behavior. And of course, every model has limitations, but could really introduce students on what modeling actually does for you, right?
Stop you for a second.
Sure.
Ingmar, I just asked you to go back to that modeling slide and could you just elaborate a little bit more about what the student would do regarding modeling? Like, we work a lot with modeling in mathematics and modeling in science. Could you just elaborate a little bit more about how the student actually interacts with the programming to develop a model?
Okay, so we have not tested that with students to let them actually model, but the way it will work, right? I mean, Scratch, you can basically draw your object that you want, like this little cell here, and then you have a few lines of code that animate and tell basically this object what to do. And in this particular case, you have a loop that runs continuously, and all it's doing is basically like move forward, so that's the speed, right? Then you have some random turning to the left or to the right, so this mimics some sort of a noise. And then we have this little loop here which is saying, like, if the light is on from some sort of direction, then you just need to change your angle relative to the light. And so this leads to the effect that when you have light from one direction, the cell is swimming this direction, that over time it will basically turn, right? So now how would a student engage with that in terms of modeling something themselves? I mean, either you can ask them to come up with all of this, or you give them something like this and only say, like, I mean, change the parameters, right? Like, you see it in the real, you see the simulation, try to match the noise, the speed, to kind of bring them in correspondence. So, yeah. And we had this on a number of science fairs and other events to have students, children basically engage with it of different ages, let them play, and also letting them fill out questionnaires. And the general thing that comes out is that, of course, children recognize and can draw these organisms. Younger kids even draw like little legs and things, so they recognize this is something living. There's also, the older kids really realize the light response. So this is what this image shows here. So when we ask them draw their swimming relative to the light, they realize that it aligns. We basically see, if you compare age groups, that around 10, 11 years, children fully get that, which actually also aligns with the school curriculum in biology. And we also asked a number of teachers about the whole features of the setup, what they liked and what they not, especially for their very own lectures that they would teach. And what they were most excited about is the fact that you can really self-build and kind of do measurements and select cells. Teachers were a little bit more divided on the usefulness of like gaming and modeling overall, but still on the positive side, but teachers have different opinions on that. And so the status of this project is basically now that we have this set up, we have tested it with teachers and with children, and we hope now to really get this out in schools to be used, to be modified, something like that. Yeah, so this is kind of the summary of that, and I think the key exciting things, I think, is that you have a system where you can really combine building something, doing inquiry, like studying biology, and then also being playful, and so combine different modalities of learning into one setup. So now let me come to a second project, which is a little bit more advanced, both into technology, but also in the way that you can interact with these cells. We again use these Euglena, and this project, as you will see, is actually one that we then brought to the museum and let people in the wild interact with that. And so the difference with this project is the following. So you have a touch screen, and you see again the same Euglena cells, and you can draw on the touch screen, but whatever you draw is basically projected as light patterns onto these cells. So you don't have just the lights from the side, you really have the lights from the top. And as this movie shows, you can basically draw a circle of light, and since the cells avoid the light, as soon as they counter it, they kind of get trapped, right? So you see, like, this cell now touches, and then it touches here, and you really trap it. And this makes for a very intuitive experience for the user to do something and to interact with these microscopic cells, okay? So here's again kind of the general idea, right? Drawing a circle, trapping. The basic setup is the following. So you have this tablet, which you then, with a projector and some right optics, not project big on the wall, but project small into the microscope in this little chamber here. We have some automatic fluid handling, so the cells are kind of living there for a couple of days to weeks in an automated way. So it's almost like plug and play, like electronics. And then there's a microscope here. We have a regular eyepiece, but also the camera that brings it back on the screen, and so we really have a closed feedback loop. Right? So this is what the setup looks in real. If you look through the eyepiece, interestingly, you also see directly the light that you project, right? And you see the cells. So these are the cells themselves. And now you can do a number of things with that. For example, simple experiments. You can ask which kind of colors or light colors do the cells respond to. So if you, for example, draw in red or green, you see that the cells don't respond, but as soon as you use the blue color, they do respond, right? And it's true that they respond more strongly to blue light in general, but we also kind of change the level such that any sort of user in a museum gets a very, very clear and robust experience, right? You can make games on this and a few other things. So we had this a couple of times in the San Jose Tech Museum and kind of testing it with what people think. And the basic setup is here's the touch screen, and then here's the setup, and we made it such that, of course, you can look through the eyepiece. You can also see the inner workings of the machine. And what this really does is it's important to have this machine next to it, because if you don't, so then many people think this is all just simulated, right? But as soon as you see the eyepiece and they draw something here and look through the eyepiece, they really start understanding the whole setup. It's also really engaging multiple people at the same time, like families, like one person is drawing something here, the other one is looking through, which then led to even kind of self-generated play, like, hey, draw something and let me guess what you do, things like that, right? And so here you see some examples. So people walk up to the system, start drawing something, just very naturally, and often only then start realizing there's actually something swimming in the background, which actually now starts reacting to whatever they draw, right? Here's a very interesting one where one person in the course of over 2 minutes actually made a little experiment. So initially, the person trapped a single cell and then made a little maze here by just erasing part of the color again, and then looked how the cell basically escaped this maze, right? And so what the system therefore enables is a person to basically make a microfluidic chip within 2 minutes without having ever done something similar like that, right? So here again, just emphasizing the fact you draw something on the screen and then actually you can see it live and small. And so then we did some further exploration in this. So this is right now collaboration with the Exploratorium. Like, what other interaction modes could we actually have? And so we used the Kinect. And so basically taking your body movements and projecting them into the microscope. And again, having it all projected on a big screen now. And we see kind of in this highly sped up movie here on the right is that, if, for example, with this picture frame, you can actually capture individual cells and kind of move the cells around. There's some other exciting aspect compared to the previous one is also that you now really see, if you look through the microscope, you see a little person moving around in there. And that's actually what children get very excited about, right? And so it's basically you meet with these cells, both big in your own world, but then you also meet with them in their small world. And so this may have some very interesting effect of how people actually perceive the interaction with these microscopic worlds. And so this is basically work in progress. And so we kind of try to understand what kind of other interaction modes are possible and what does it lead to in kind of informal learning spaces like a museum. Right? So here's another example where we use basically the same organisms but on a much kind of zoomed out scale and in many more organisms, right? So right now you always saw like a single cell that you basically interacted with. Here's something where we have 1 million cells. So you see this little micro aquarium here, which is basically a closed disk with these Euglena cells inside. So it's about millions of them. And if you shine light from below, then they all swim up and then even start some bioconvection patterns. And now if you shine some sort of a image from below, then you can actually make some sort of a photograph, which then later develops into something more dynamic. So what you see here on this big screen is basically you shine this picture on, and you see wherever there's light, that's where the cells start moving first and fast, and then over time it kind of develops further, right? And so it's just another way on a very different length scale to interact with these cells. Now you can really see these patterns by eye. You can't really see the individual cell anymore, right? Okay, so those were the kind of more direct interactions. Yeah.